Hello all, welcome to our channel Polyhipster. In this video, we will be discussing about one of the main group of cell surface receptors known as the G protein coupled receptors. So let's begin. To introduce, the G protein coupled receptors are the largest family of cell surface receptors. The action of the GPCRs depends on the receptor, the G protein, and the effector molecules. So as we can see in the diagram, the ligand comes and binds to the receptor part of the G protein coupled receptor, which is attached to the G protein and which are attached to three different effector molecules. Now these receptors are hydrophilic in nature. Few examples include the catecholamine receptors, which include the epinephrine, norepinephrine, and dopamine receptor, the acetylcholine receptors, glucagon, serotonin, secretin, pituitary hormone receptors such as TSH, ACTH, FSH, and LH. Now let's talk about the receptor first. The receptor is a 7-transmembrane helix receptor. It has 7-transmembrane helices as shown in the diagram. The outer surface binds to the ligand, which is extracellular, and the inner surface, which is intracellular, binds to the G protein. Next, let's talk about the G protein or the guanine nucleotide binding protein. It is a heterotrimeric protein, that is, it has three subunits alpha, beta, and gamma subunit. To look into details, the main function of alpha subunits is to activate GDP to form GTP, that is guanosine diphosphate, guanosine triphosphate. GTPase, which is a small G protein that functions independently, inactivates or terminates the activity of alpha subunit by converting GTP back into its inactive form GDP by the process of hydrolysis. Now next we go to beta subunit. The main function of beta subunit is to form a stable complex with gamma subunit. Similarly, the main function of gamma subunit is to form a stable complex with beta subunit and with a lipid anchor in the cell membrane. So how are these G protein coupled receptors activated? Let's see. So first, the hydrophilic extracellular ligand. It could be a hormone or any molecule that activates a G protein coupled receptor. It comes and it is recognized by the receptor and therefore it gets bound to the receptor, which causes a conformational change. That is, the receptor is now activated. The receptor binds to the G protein extracellularly. Now the G protein gets activated and binds GTP instead of GDP. So the alpha subunits get activated now and it dissociates from the beta gamma complex. The alpha subunit now activates the effector molecules, that is the GQ, GS, and GI. The function of GQ being activating the phospholipase C, which increases the calcium inflow. GS, it activates adenylate cyclase and it causes increased activity of cyclic AMP. Whereas GI decreases the adenylate cyclase and therefore it causes decreased function of cyclic AMP. So how does cyclic AMP act? So ATP, which is adenosine triphosphate, which is the energy currency in our body, can be converted into cyclic AMP, that is cyclic adenosine monophosphate, by the enzyme adenylyl cyclase. This reaction occurs in the cytosol and plasma membrane of human cells. What is its significance? Cyclic AMP is important in various human health and diseases. It is important in the olfactory system where it helps convert odor chemicals to neural signals. Now, this cyclic AMP, when acted upon by phosphodiesterase, it is converted into degradation products. Now, another scenario is where ATP can be converted into ADP by the loss of an inorganic phosphate in the presence of ATPase enzyme. The remaining ATP can be converted into cyclic AMP, which in increased concentration of cyclic AMP can activate protein kinase, which leads to further reactions. For example, let's talk about muscarinic receptors, which are parasympathetic receptors. They are activated by GQ coupled effector molecules. It proceeds by activating the phospholipase C, which splits PIP2, which is phosphatidyl inositol 4,5-bisphosphate, which is a signaling lipid that is involved in many cellular processes. PIP2 is split into IP3 and DAG, which are inositol triphosphate and diacylglycerol respectively, which causes increased influx of calcium molecules into the cell, leading to the action of smooth muscle contraction and activation of protein kinase C. Few examples of the receptor where this procedure is seen is the thyroid releasing hormone, gonadotropin releasing hormone, V1 receptor of ADH, oxytocin receptor, H3 receptor of histamine, angiotensin 1 and gastrin. Next, we'll talk about the beta adrenergic receptors, which are sympathetic in nature and activated by GS coupled effector molecule, which acts by activating the adenylyl cyclase enzyme, which converts ATP into cyclic AMP, which in turn activates protein kinase A. And protein kinase A acts in the following way. First, it increases the calcium influx, especially in the beta 1 receptor, which is present in the cardiac muscle, thereby increasing the cardiac contractility. Next action is inhibition of myosin light chain kinase in the beta 2 receptors, which causes increased bronchodilation, increased vasodilation, and tocolysis, which is the relaxation of uterine muscles. Next, it acts on the kidney beta 1 receptor by activating renin, which activates angiotensin 2, which releases aldosterone, which helps to regulate blood pressure, sodium potassium levels, and blood pH. Last, it acts by increasing aqueous humor secretion in the eye. If we see the sympathetic system, which is mainly concerned with the fight, flight, and fright response, we can see that the main substrate here is phenylalanine, which is an amino acid. In the nerve ending, phenylalanine is converted into tyrosine, then L-dopa, dopamine, and norepinephrine. Finally, epinephrine, which is released from the nerve endings into the NM junction to form a certain reaction. Sympathetic receptors are mainly of two types, alpha and beta. Alpha can be divided into alpha 1 and alpha 2, whereas beta into beta 1, beta 2, and beta 3. Alpha 1 receptors are activated by GQ coupled effector molecules, whereas alpha 2 is GI coupled, whereas all beta receptors are GS coupled receptors. Alpha 1 is pro-sympathetic, that is, it increases norepinephrine release, whereas alpha 2 is anti-sympathetic, it decreases norepinephrine release. Whereas the parasympathetic system, which is mainly consisting of the muscarinic receptors, they are of five types, M1 to M5, out of which M1, M3, and M5 are GQ coupled, whereas M2 and M4 are GI coupled. The GQ coupled receptors, they increase the calcium inflow, leading to contraction of smooth muscles and secretion from the glands. Whereas the GI coupled, which is M2 and M4, they cause inhibition of these parasympathetic see how these muscarinic receptors act in various parts of our body, starting with various glands. In the glands, M1 and M3 receptors are mainly present. One acts along with the submucosal plexus to increase the gastrointestinal secretion, whereas M3 increases the secretion of tears, saliva, bronchial secretion, and sweat. In the eye, we mainly have M3 receptor, which causes increased contraction of sphincter pupillae, leading to miosis, and increased contraction of ciliary muscle, helping in accommodation. In the heart, we have M2 receptors, which decreases the rate of firing from sinoatrial node and atrioventricular node, which in turn decreases the heart rate, leading to bradycardia. In the lungs, we have M3 receptors, which increases contraction of bronchi, leading to bronchospasm, and as discussed before, increase bronchial secretions from the glands. In the GI and GU system, we have M3 receptors mainly present, which increases the contraction, absorption, motility, secretion, digestion, and void. Also, increased muscle contraction and sphincter relaxation. So, the nitric oxide, which mainly acts on M3 receptors, it causes muscle relaxation and vasodilation. Finally, let's talk about the dopamine receptors D1 and D2. D1 activates direct pathway at striatum. It causes vasodilation in the kidney and it is GS coupled. Whereas D2 receptors inactivates the direct pathway at striatum and it is GI coupled. The H2 receptors, histamine receptors, H1 is GQ coupled, it increases the calcium influx and causes bronchoconstriction. Whereas H2 receptors are GS coupled, it increases activation of cyclic AMP and causes increased gastric acid secretion. That's all for now. See you soon with more details and further exciting topics.